Welcome to the second video in our first series of videos. In this discussion, I'm going to cover some of the most important ideas governing the development of the field of biochemistry. A central theme in this course is the explosion of the idea that knowledge is a fixed entity, not subject to change, and this idea of the central dogma of biology is a nice example of that. Some of you have probably heard of the term central dogma before, and just the fact that we use the term dogma to describe this idea gives you the sense that it's a little bit maligned nowadays. In any case, the central dogma of molecular biology is that DNA makes RNA, and RNA makes protein. And that's what these bold arrows represent in this diagram. We see a DNA molecule which encodes for all of our genetic information being converted into RNA. In this case, we're looking at happens to be a piece of messenger RNA, and that messenger RNA goes on to carry some information across via the ribosome into the synthesis of a protein, which then goes off and does its job. This is a very simplistic idea and is constantly changing and developing, and that's what these dashed arrows are supposed to indicate for you. First of all, in terms of DNA and RNA, RNA actually goes back and interacts with DNA as well, and that works through the silencing of the genetic material and the silencing of the transcription of certain kinds of genes. I won't go into greater detail than that right now, except to just restate that RNA can go back and alter the behavior of DNA as well. Again, too, it's a little more complicated than simply RNA making protein. In this arrow here, I'm indicating that maybe the RNA actually does something to itself too, and that gives you this idea of the ribozyme. So an enzyme is a protein machine, carries out some chemical catalysis. A ribozyme is a ribonucleic acid catalytic machine, and in this case, we'll see an enzyme that can act on itself or auto-catalyze some chemistry, but as well we'll see that these ribozymes can carry out catalysis on other molecules as well. So RNA is a little more complicated than RNA simply making protein. Now, things get more complicated still. Intermolecular interactions affect the structures of both partners. So intermolecular, that is proteins interacting with each other or proteins interacting with RNA or DNA, affect the behavior of those molecules through the changing their structure. There's some interesting examples of this. Going back to the idea of RNA making protein, normally we might think of RNA as being this thing that carries the genetic information from DNA all the way out to making the protein, and we'll see that there are a few different kinds of RNA molecule, but the RNA isn't just an encoder or a transcript of the DNA. It can be a machine, and in the case of the ribosome, it's a machine that carries out the synthesis of proteins. And the RNA itself is an important part of that machine, but you don't have a ribosome without protein too. So the protein models the structure of the RNA, and the RNA can then go ahead and catalyze the synthesis of proteins. So RNA makes protein is a little bit simplistic. Another great example of how DNA can affect the structure of proteins and proteins can affect the structure of DNA is the modeling of the chromatin structure. So you probably are aware that DNA is organized into chromosomes, and we refer to the organized structure of DNA, for example, in a eukaryotic nucleus, as the chromatin. And the structure of that chromatin is really important in terms of how the DNA gets used in living systems. Well, the structure of the DNA, in this case the methylation state of the DNA, can drastically affect the structure of the chromatin, altering the ability of the DNA to be read, for example. The structure of proteins that interact with DNA, the histones that are the central organizer of the chromatin in the nucleus, can also be methylated, and that can affect the organizational state of the DNA. So detailed structural changes in DNA can affect the way the DNA interacts with protein and vice versa. So you have a crosstalk all the way around this circle. So now we see that this really is a circle and not just a one-way flow of information from DNA to protein. That's the development of this idea that the complexification, if you will, of the idea that DNA makes RNA makes protein. It's a much bigger issue than that. Let's look at a little timeline here at the development of biochemistry, and this is a nice breakdown of sort of a trajectory of scientific problem solving, if you will. The way I like to think about this is, beginning in the Renaissance, 1600s, all the way up to the present, what we see is a sort of a trajectory where we go from observation to the synthesis, and in this case, I mean synthesis both in terms of the synthesis of ideas, or the collation, if you will, of all of those observations into a useful theory, perhaps, and I also mean the actual physical synthesis of molecules as well. That synthesis of molecules, of course, leads us to this idea, or this fact, now that science is focused on the manipulation of objects, not just the observation of the function of those objects or the roles of those objects, but also the harnessing of those objects to our own ends. There's a couple of milestones that are interesting, so I'm just going to relate those to you. First of all, the development of microscopy. Here I've got two names, Van Leeuwenhoek, who is a Dutch scientist, and then Hooke as well. Both of these guys, Dutch, and then Hooke, Robert Hooke being English, both of these guys playing important roles in the development of microscopy. The disprovement of spontaneous generation. I'm going to attribute this to Redi, who is an Italian, polymath. There are other people that were working on this around the same time, and ultimately Pasteur put the kibosh on the idea of spontaneous generation. But Redi did an interesting experiment wherein he proved that life does not emerge from nothing by covering a few different jars, or in which there was some rotten meat or something, and some of the jars were open and some of the jars had a fine mesh covering them up, and of course in the jars that were open, flies could fly in and maggots emerged, right? So up to this time, and the notion that Aristotle put forward, that the maggots emerged on their own out of nothing, that had been the prevailing knowledge, the prevailing belief, right? But now Redi has actually done an experiment in which he shows that where the jars are covered up and the flies can't enter, no maggots emerge until you take the cover off, and then of course you get maggot formation. So this is an experiment, right? So even now we're turning at this period of time away from the simple notion that knowledge can be received from the ancients, and then instead we test the boundaries of our knowledge through experimentation. Moving forward in time, so here I say plants convert CO2 to oxygen. Joseph Priestley did a great experiment in which he had a covered jar, burned a flame, saw that the flame went out when the oxygen was consumed, what we now refer to as oxygen, and then that oxygen can be replenished with the presence of a plant. So chemical conversion carried out by a plant. Now I put the cross through there, or that X on that because that's a dramatic oversimplification. Those of you that have been through some of your basic biology classes are going to know that the fixation of CO2 and the production of O2 are separated chemical reactions, but we'll conflate them for our simplistic purposes here. Just realize that I'm not trying to mislead you, but I'm trying to move quickly through this timeline. So that's an important observation. Plants perform chemical conversions. We can do some of those chemical conversions ourselves. Friedrich Wöhler, German chemist in the 19th century, completes the synthesis of urea through the Wöhler reaction. I'm not going to discuss the chemistry there. You can refer back to organic for that. But that's, again, the notion that we now are gaining control over our chemical components, and we can do with them what we will. Buried in here too is The Origin of Species. I have the publication of The Origin of Species. I'm going to always throw that into a timeline of biochemistry. I'm going to talk more about Darwin in a few moments, so I'll leave it there as an obvious player in the history of biochemistry. Instead, let's think about chirality and Pasteur's role in the development of the notions of chirality. Again, now, Pasteur at this point making an observation of physical differences between chemically identical compounds, tartaric acid isomers. And then jumping forward, we see Emil Fischer, again a German, developing a nomenclature for stereochemistry. Now, I want to hasten to add that he wasn't the developer of stereochemistry, and really we can even leap back to Pasteur if we really want to assign some primacy in terms of the development of those ideas. But he's presenting a nomenclature or a system of talking about stereochemistry. So we can go from here, from the observations, to the synthesis of knowledge into a workable framework. And there in the middle somewhere we have Kekulé, again another German, who describing the structure of benzene as a snake biting its tail. I'll refer you back to your organic chemistry course for a reiteration of that story. Let's leap ahead into the 20th century, though, where we begin to develop a good enough knowledge basis to begin manipulating molecular biology. So James Sumner in 1928 purifies an enzyme and identifies that enzyme as an actual protein. And he gets a Nobel Prize for that a little bit later, so that's a milestone. Of course, in 1955, we get the structure of DNA with Watson and Crick, and some protein structures are being discovered around the same time. We won't go into that in this lecture any deeper. But again, an obviously important milestone. But in terms of manipulation, the discovery of restriction enzymes that cleave DNA at very specific locations, we attribute to Nathan Zarber and Smith, who received a Nobel Prize in 1978 for their work earlier in the 70s at identification and purification of those restriction enzymes. And of course, PCR invented in the late 20th century. I'm going to talk about Kary Mullis as well, but in both of these two cases, restriction enzymes and in PCR, we are now at the point of taking biological hardware out of context and using it for our own human objectives. So that's, I think, a really interesting trajectory. Let me talk for a few minutes about some historical figures, and I've mentioned a couple of these already in the previous slide. And again, what I really want to emphasize here is that is scientific method, right? So we develop hypotheses based on observations, and we design and carry out experiments to test those hypotheses. And finally, we develop models and theories based on the outcomes of those experiments and the acceptance or rejection of our hypotheses. So Louis Pasteur, active in France, late 19th century, late 19th century, quite a laundry list of accomplishments, and I don't want to go through all of those, but I think some of these highlight this idea that at this point in history we're starting to move from simple observation to action, right? So to actual utility of knowledge. So he observes that microorganisms select a single enantiomer in racemic solutions. So that's, again, getting back to this idea of chirality, and this is going to be something important in our discussions of enzymes as well. Enzymes are chiral systems, and they can recognize, and most often do recognize, individual isomers and discriminate based on chirality. He also makes a really important observation, for those of you that like to brew beer, for example, that yeast and bacteria carry out different functions in fermentation, and it's important to segregate those activities if you want to get something that doesn't taste terrible. He also finally puts the nail in the coffin of spontaneous generation. That's something, again, that was being bandied about for over 1,000 years up till he came along. And then, of course, he's responsible in terms of putting ideas into action for the idea of pasteurization and sterilization and disinfection of foodstuffs. Pasteurization, of course, is raising something, for example, milk to 55 degrees centigrade to kill various microorganisms. And, of course, the development of vaccines, which we're not going to go into, but, of course, emphasize the power of application of the models that you develop based on your observations. Charles Darwin, this again, the theory of evolution, or the model of evolution, the theory of natural selection, is so central to biochemistry that I can't avoid mentioning this individual's name. Again, though, what I want to emphasize is not so much the idea of evolution now, but the fact that useful models make predictions that you can test. And this is something that's so wonderful about the notion of evolution, is that it provides us for a framework for thinking about biology, which allows us to probe and deepen our knowledge, as opposed to just presenting us with a set of received knowledge or facts that we can't really probe. Of course, he was acting as a naturalist on the HMS Beagle in the middle of the 19th century, and on that tour made a series of wonderful observations about the diversity of life that helped him develop his idea of natural selection. Jumping ahead, I want to talk about Chris Anfinsen. We'll spend much of our time in this class discussing structural biology, and he plays such an important role in the development of a sort of a reductionist view. And again, he's a Nobel laureate, wins the Nobel Prize in 1972, again for work that he was performing a little bit earlier in his career. What Chris Anfinsen does is apply physical methods to biological systems, right? So we have a new set of tools that we can apply to ask new questions about biological molecules. The cool thing that he does is he observes reversible denaturation. So this is a complicated idea, for those of us that haven't studied protein structure yet. But what I want to emphasize is that proteins have structures that we can destroy, for example, by raising the temperature or changing the pH or something like that. What he observed is that we can not only destroy, but we can recover the biological activity by an enzyme, and that's it's so hard to overemphasize the importance of that fact, because what it implies is that there's nothing special about some vital force in a living system. What governs the function of the molecule is its structure. When you remove that structure, you can get it back just by reversing the conditions, and that implies that the function of the molecule is embedded in its amino acid sequence. And again, if that's an idea that's alien to you, stay tuned, because we're going to spend the next 3 weeks really clarifying that. Again, the idea, though, is that he links structure and function to amino acid sequence. I got two more sort of biographies to throw at you. First of all, Rosalind Franklin. The reason for showing this slide of Rosalind Franklin is not only to say that science is a human endeavor, and that means that there are hiccups along the way that have everything to do with the failure of human behavior and less to do with the failure of science with a capital S. So let me just, for those of you that are not familiar with the story, there's a whole group of structural biologists doing crystallography to study the structure of DNA in London and other molecules as well, working in London at different universities in the early 50s. Rosalind Franklin was one of them. She collected important structural data on DNA using X-ray crystallography. Now, ultimately, it was model building based on that data that she collected that led to a solution of the structure of DNA, and which was published in Nature in 1955. But Rosalind Franklin wasn't attributed. And those of you that have done some research on your own, I guess I'd like you to think about how you would feel if someone used your data to publish a landmark paper in all of science, let alone biology, without proper attribution. So now there's a lot that's been discussed on this subject. I'm not going to go more into it, except to say that perhaps bad feeling between all the authors involved might have led to Franklin's exclusion from this ultimate structural solution. Now, of course, Nobel Prizes were awarded to a bunch of individuals for the solution of the structure of DNA. Rosalind Franklin was dead at that point, and therefore excluded from Nobel Prize. But she certainly would have been awarded the Nobel Prize along with those sort of naughty teammates, if you want to think of it that way. So there are problems in science, but hopefully we trundle on. Moving along, though, to Kary Mullis. He's still alive. I think he's surfing somewhere down in San Diego right now. I don't know. The machinery of living systems can be turned to human goals. So this was, I said, if we want to think about biochemistry as following a trajectory leading from the development of a set of observations into a theoretical framework that allows us to ultimately turn the objects of our study to our own ends. The invention of the polymerase chain reaction, which Kary Mullis is responsible for, is a super example of this. So he's working as a consultant, synthesizing DNA molecules in 1979. His breakthrough was to think that, well, there's this enzyme in all biological systems called polymerase, and what it does is replicate DNA. Why can't, instead of chemically synthesizing these oligonucleotides myself, why can't I use the polymerase to do it? It's been optimized for that job over billions of years and successfully amplifies DNA by many, many orders of magnitude. So what he does is develop this system and prove that it can work. And now, of course, polymerase chain reaction is at the heart of so many molecular biological methods and is probably familiar to all of us. And if it's not now, it certainly will be by the end of the next 15 weeks or so. And he gets a Nobel Prize for his work in 1993. So I hope with this brief overview of the history of biochemistry, what I've given you the idea of is that knowledge is something that is constantly in development and is being carried out by human beings that are working with their own set of pressures and difficulties, and to ultimately, though, to now bring us to the point where we can use biological systems to not only to answer questions about biology, but to solve human problems as well. And with that, we're at the end of the second video. Next up, video 3, in which we'll be looking at the basics of cellular organization.